Welcome back.
Whether you are frantically trying to catch up on the latest pharmaceutical trends for a big meeting, or you're just insanely curious about the drug that absolutely everyone seems to be talking about right now, you are in the exact right place. Welcome to this deep dive.
Yeah, thanks for having me. It's definitely the topic of the decade.
Oh, 100%.
Yeah.
Today, we are mapping out the medical history and well, the wild market dynamics behind GLP-1 receptor agonists.
Which you probably know better by brand names like Ozempic or Wegovy.
Right. And our mission today is to figure out how studying deep-sea creatures and venomous lizards somehow led to the biggest blockbuster drug of the 21st century.
That sounds like science fiction,
It
honestly.
really does.
Yeah.
Okay, let's unpack this. To get to the core of the mystery, we first have to ask, why did we even need this drug in the first place? I mean, we've had insulin for a long time, right?
If we connect this to the bigger picture, to really understand the origin of a drug like Ozempic, we actually have to travel back to the post-World War II era. As modern diets and lifestyles began to shift globally, the rates of type 2 diabetes started rising significantly. And you're right, doctors already had standard insulin. It was discovered back in the 1920s and was a literal lifesaver for type 1 diabetics. But standard insulin just wasn't enough anymore.
Because type 1 and type 2 are, they're fundamentally different problems, right?
Exactly. Type 1 diabetes is a lack of insulin production. The factory's broken. But type 2 is characterized by insulin resistance.
Got it.
Your pancreas is still releasing insulin, but your body's tissues, your muscle and fat cells, they just aren't responding to it. They aren't taking in enough glucose from your bloodstream.
So it's like having the right key, but the lock is jammed.
That is exactly it. So what does the pancreas do? It senses the high blood sugar and desperately pumps out even more insulin to compensate. It's trying to force the door open.
Which sounds exhausting for the body.
It is. Over time, that relentless cycle burns out the insulin-producing beta cells in the pancreas until they eventually fail. A completely new medical strategy was desperately needed because simply giving a patient more insulin wasn't fixing the underlying resistance.
And that search for a new strategy led to a totally surprising observation in the 1960s. There was this amazing experiment involving how we consume sugar.
Yes, the glucose tolerance tests.
Right. So if you give a patient glucose by putting it straight into their blood intravenously, their blood sugar goes up and their insulin goes up a certain amount. But if you have that same patient drink the exact same amount of glucose, you get a massive, much larger insulin spike.
Which is a profound realization when you think about it.
Yeah.
The total amount of sugar entering the body is identical, but the delivery method changes the body's reaction entirely.
Right. It meant the gut itself was somehow tasting or processing the glucose and sending a secret, amplified signal to the pancreas, telling it to secrete more insulin. They coined a term for this gut signal, an incretin hormone.
And once that incretin effect was identified, scientists immediately started hunting for which specific proteins in the gut were acting as these messengers.
Because if they could isolate it, they could use it.
Precisely. If they could isolate the incretin, they could potentially turn it into a powerful new diabetes treatment. One of the very first potential matches they started looking at was a hormone called glucagon.
Which is often described as insulin's buddy cop.
I like that analogy.
Yeah, they both come from the pancreas, but they do opposite jobs to keep your blood sugar perfectly balanced. Insulin lowers blood sugar when it's too high, and glucagon raises it when it's too low.
Right.
But researchers in 1965 found something incredibly counterintuitive. If you injected someone with glucagon, their insulin levels increased before their blood sugar even went up. The glucagon itself was directly stimulating the insulin secretion.
Exactly. It was acting like the incretin they were looking for. But there was a massive roadblock. Studying these specific hormones in mammals is incredibly difficult because the mammalian pancreas is intensely crowded. Well, the vast majority of a human or mammalian pancreas is made up of what are called acinar cells.
And those just make digestive enzymes, right? They don't have anything to do with insulin.
Precisely. They are factories for digestion. Scattered amongst all those millions of digestive cells are tiny, microscopic clusters called the islets of Langerhans, and that's where the insulin and glucagon are actually produced.
Wow, okay.
To give you an analogy, trying to study glucagon production in a human pancreas is like trying to study a few specific rare trees hidden deep inside a massive, incredibly dense jungle. It was a biochemical nightmare to isolate what was happening.
Okay, here's where it gets really interesting. Because the mammalian pancreas was too crowded, too dense to study easily, Scientists in the 1980s realized they had to look completely outside the human body. They turned to the anglerfish.
Yes, the deep-sea fish with the bioluminescent lure on its head.
It sounds crazy, but it turns out the anglerfish is biologically unique. Unlike humans, it has entirely separate organs for making digestive enzymes and making hormones. The jungle was separated from the trees.
Which is a researcher's dream.
Exactly. So scientists started looking at anglerfish DNA to understand how a body actually builds these hormones from scratch.
And what they saw in the anglerfish fundamentally changed our understanding of endocrinology. They discovered that the anglerfish doesn't just produce a tiny glucagon molecule directly. It starts with a massive precursor protein called pre-pro-glucagon.
Think of this massive precursor protein like a sheet of notebook paper with several perforations punched into it. The cell's machinery is designed to tear the protein apart exactly at the specific cleavage sites.
That's a perfect way to visualize it. The cell uses enzymes to chop that large inactive protein at those perforations, breaking it down into smaller functional pieces.
Right.
And when scientists looked at all the pieces left over after the cuts were made, they found the glucagon they expected, but they also found a totally new, previously undiscovered peptide. They called it a glucagon-related peptide.
So the obvious next question is, do humans have this too? Fast forward to 1983. Scientists take this anglerfish discovery and apply it back to mammals, specifically using hamster DNA to see if we have those same perforations and the same hidden peptides.
And we do. They found the mammalian versions and named them GLP-1 and GLP-2 glucagon-like peptides.
Boom. Mystery solved.
Well, you would think. But science is rarely easy. There was a major hurdle right out of the gate. When they synthesized and tested the full 37 amino acid version of GLP-1 that they found in the DNA, it did absolutely nothing.
Wait, really?
Yeah, it didn't stimulate insulin secretion at all. It seemed like a total dead end.
Which brings us to a brilliant scientist named Svetlana Mojsov. In the mid-80s, she was studying GLP-1 alongside Joel Habner, who is one of those original anglerfish researchers. Mojsov looked at the chemical structure, looked at the whole 37 amino acid chain, and made a crucial deduction.
It really was a stroke of genius. She noticed that the full 37 amino acid chain didn't quite look right. But if you remove the first 6 amino acids from that chain, the remaining peptide looks structurally identical to active glucagon.
Just a slight trim.
Exactly.
Ah.
She hypothesized that the cell wasn't stopping at the first set of perforations. It was actually making one more crucial cut, cleaving off those first 6 amino acids. She believed this shorter remaining portion, which became known as GLP-1 737, was the true biologically active hormone.
And in 1987, she proved it. She demonstrated that this exact shorter version was the actual key that fit into the lock to stimulate insulin release in the mammalian body. It was a monumental breakthrough for diabetes research.
Huge.
But as with all great medical discoveries, there was a catch.
A very frustrating catch.
Right. On one hand, GLP-1 has this incredible built-in safety mechanism. It only raises insulin when your blood sugar is elevated. If your blood sugar is normal, it doesn't do anything.
Which is fantastic because it helps patients avoid dangerously low blood sugar, known as hypoglycemia.
Which is a major risk with traditional insulin injections.
Yes.
But the catch is its lifespan. How long does native GLP-1 actually last in the human bloodstream?
About 2 minutes.
2 minutes.
Yes, 2 minutes. The human body is incredibly efficient. As soon as GLP-1 is released and does its job, an enzyme called DPP-4 swoops in and chops it up. And then the kidneys filter it out almost immediately.
That's just wild.
It is. And you simply cannot make a daily take-home medicine out of a molecule that disappears in the time it takes to brush your teeth. The only way it would work is if a patient was permanently hooked up to an IV drip in a hospital.
So we have a miracle mechanism that vanishes in 120 seconds. This sets the stage for a massive global race in the pharmaceutical world. How do we make this hormone last longer? And two completely different competing solutions emerged.
Let's look at the American solution first, because it relied entirely on nature, and honestly, it is one of the strangest origin stories in modern medicine.
I couldn't believe this part of the research. In the late 80s and early 90s, a researcher in New York named Dr. John Eng was looking at something that had absolutely nothing to do with diabetes. He was studying the venom of the Mexican beaded lizard and the Gila monster.
Right, the desert lizards.
Yeah. These are venomous lizards native to the American Southwest, and they are known for having intensely painful bites, but also very, very slow metabolisms. Eng isolated a specific peptide in the Gila monster's venom called exendin-4.
And the chemical structure of exendin-4 was a revelation. It structurally mimicked human GLP-1 closely enough that it's successfully bound to our mammalian GLP-1 receptors. It fit perfectly into the human lock.
Okay.
But because it evolved in a desert lizard, human enzymes like DPP-4 didn't recognize it. They couldn't chop it up. So instead of lasting 2 minutes, this lizard peptide lasted for hours in the human body.
It's just wild that the answer to human metabolic failure was hiding in reptile venom. Eng eventually licensed this discovery to a biotech company called Amylin, and by 2005 they had developed it into a synthetic drug called exenatide, which was FDA approved under the brand name Bieta.
Right.
It was a massive leap forward, a twice-daily injection for type 2 diabetes. But at the exact same time, a totally different approach was being cooked up over in Europe.
Right. Over in Denmark at the pharmaceutical giant Novo Nordisk, they don't look to nature. They look to bioengineering.
Yeah.
In 1994, a scientist named Lotte Bjerre Nedsen found herself in a uniquely high-pressure situation.
It's a ticking clock.
Exactly. After a corporate restructure, she was essentially the only scientist left on her team pushing the GLP-1 project. The company's executives were heavily focused on their traditional insulin products. The CEO literally gave her a 12-month ultimatum, come up with a viable, long-lasting GLP-1 drug, or the entire project gets permanently scrapped.
Talk about pressure. So how did her team solve the 2-minute problem without using lizard venom?
They used a brilliant molecular design trick. They took the human GLP-1 peptide and attached a fatty acid chain directly to it.
Why a fatty acid?
Because fatty acids love to bind to a protein called albumin. Albumin is the most common protein in human blood plasma. You can think of albumin like massive cargo ships constantly circulating through your bloodstream.
I love that.
By attaching that fatty acid chain to the GLP-1, Newson's team essentially gave the medicine a grappling hook. The GLP-1 molecule attaches itself to the massive albumin cargo ship.
Making it a much chunkier molecule overall.
Exactly. And because it's now riding on this giant protein, the kidneys simply cannot filter it out. It's too big to pass through the cellular filtration system quickly. It slowly detaches over time, providing a steady release of the hormone.
Oh, so elegant.
It really was. This designer approach resulted in a drug called liraglutide, branded as Victoza. Thanks to that albumin trick, it had an 8 to 10 hour half-life, which meant patients only needed one single dose a day.
So what does this all mean for the patient? It means we suddenly have highly effective, convenient diabetes drugs.
Huh.
But during the animal trials for liraglutide, the researchers started noticing a very strange, very consistent side effect.
Yes.
The animals were eating significantly less. In fact, some were severely suppressing their appetites to the point where they were practically starving themselves. Knudsen saw this data and immediately wondered the obvious: could this highly effective diabetes drug actually be a weight loss drug?
But when she brought this idea to the executives, the CEO of Novo Nordisk was incredibly reluctant. He actually pushed back, famously stating at the time that obesity was a social and cultural problem, not a medical one.
Which sounds so dismissive now.
It does, but to be fair to him, you have to look at the historical context of the pharmaceutical industry to understand his deep hesitation. The history of weight loss drugs was a graveyard of medical disasters.
It really was. I mean, the very first FDA-approved weight loss drug back in 1947 was desoxyephedrine, which is quite literally methamphetamine. It suppressed appetite, sure, but it was highly addictive speed.
And that dark history didn't stay in the mid-century. It continued right up into the late 1990s with the fen-phen craze.
Oh, wow, fen-phen.
Yeah. Fen-phen was a wildly popular combination of two appetite-suppressing drugs. Millions of people took it, and it caused massive weight loss. But by 1997, the medical community realized it was linked to severe, irreversible heart valve damage.
That's terrifying.
It was. The FDA had to step in, the drugs were aggressively pulled off the market, and the manufacturer faced billions of dollars in catastrophic lawsuits. The entire pharmaceutical industry was deeply spooked. Nobody wanted to touch an anti-obesity medication for fear of repeating the fen-phen disaster.
But eventually, the sheer scale of the global health landscape forced a shift. By 2011, data showed there were over 500 million people worldwide living with obesity. It was an undeniable crisis. And then a major turning point happened in 2013. The American Medical Association officially recognized obesity not as a moral failing or a lifestyle choice, but as a complex, chronic disease requiring rigorous medical intervention.
That classification changed everything. It provided the medical and ethical cover the industry needed. Novo Nordisk pivoted. They recognized the massive clinical need, and they took their daily diabetes drug, liraglutide, ran new trials, and the FDA officially approved a higher 3 milligram dose, specifically for weight loss in 2014, under the brand name Saxenda.
And at this point, the actual mechanism of how it caused weight loss was becoming a lot clearer. It wasn't just fixing insulin resistance in the pancreas.
Not at all. Researchers mapped exactly where these GLP-1 receptors were located in the human body. Yes, they're in the gut, but they're also deeply embedded in the nervous system, specifically in the brainstem and the hypothalamus.
So it's going right to the brain.
Exactly. The drug crosses the blood-brain barrier and sends powerful, persistent signals of fullness directly to the brain's appetite control centers.
It's literally telling your brain, hey, we just ate a massive meal, even if you haven't.
Precisely. And simultaneously, it works on the stomach itself by significantly slowing down what's called gastric emptying. The muscles of your stomach literally hold on to food longer, slowing down digestion, which creates a physical sensation of being full to match the chemical signal in the brain.
It's a powerful 1-2 punch, but there was still a sticking point. Saxenda was a daily injection. And taking a needle every single day is a huge barrier for a lot of people. So the race was back on how do we create a once-a-week injection.
Novo Nordisk went back to their albumin binding strategy. They took the GLP-1 peptide and modified it further, attaching a much longer 18-carbon fatty acid chain. This allowed the molecule to bind to albumin even tighter and resist those dissolving enzymes even longer.
And that became semaglutide.
Yes, this new, highly resilient molecule was semaglutide.
And semaglutide worked brilliantly as a weekly dose. But before it was even approved for weight loss, there was a massive, highly influential study published in 2016 regarding its use in diabetic patients. It didn't just lower blood sugar, it drastically reduced the rate of major cardiovascular events, heart attacks and strokes.
It was an absolute paradigm shift, a drug that protects the heart while treating diabetes. They branded this weekly injection as Ozempic. But the doctors running those cardiovascular trials couldn't ignore the incredibly obvious side effect. Their diabetic patients were regularly losing 12 to 17% of their total body weight.
Which is staggering. That level of weight loss was totally unprecedented for a safe, FDA-approved medication. So Novo Nordisk ran the obesity trials, and by 2021, the FDA approved a higher dose of semaglutide specifically for weight loss under the brand name Wegovy.
Right.
But they weren't the only players in the game anymore. The American pharmaceutical giant Eli Lilly was engineering their own powerhouse.
Yes. Eli Lilly developed a molecule called tirepatide, which they eventually branded as Mounjaro for diabetes and Zepbound for obesity. And tirzepatide is fascinating because it's not just a GLP-1 drug. It's what's called a dual agonist. It targets the GLP-1 receptors, but it also targets a second incretin receptor in the body called GIP.
Okay, so what does GIP do differently?
GIP stands for glucose-dependent insulinotropic polypeptide.
That is quite the name.
It is a mouthful. It's another hormone naturally produced in the gut that also stimulates insulin. But when you combine it with GLP-1 into a single molecule, they act synergistically. The GIP seems to enhance the metabolic effects and significantly reduce some of the gastrointestinal side effects like nausea that people get with pure GLP-1s.
That makes sense.
The clinical results of this dual approach were staggering, with trial patients routinely seeing over 20% body weight loss. We are talking about surgical-level weight loss in a weekly pen.
And right as these incredibly powerful drugs, Wegovy and Mounjaro, are getting FDA approval, we hit an absolute cultural explosion.
Yeah.
TikTok. The hashtag Ozempic went from around 2 million views in 2021 to over 1 billion views in 2023. Hollywood celebrities were rumored to be taking it. Influencers were documenting their weight loss journeys daily. People weren't talking about this complex peptide hormone like a sterile medical treatment. They were treating it like a viral consumer tech product or a trendy skincare routine.
And that unprecedented viral demand collided head-on with a massive supply chain disaster. In late 2021, right as the hype was building, the FDA actually shut down a key third-party manufacturing plant in Europe that Novo Nordisk relied on for their injection pens due to severe quality control issues.
So you combine infinite, culturally driven demand with a crippled manufacturing supply chain, and you get exactly what you'd expect. Massive global shortages.
Yeah.
People who genuinely needed the medication for diabetes couldn't find it anywhere.
What's fascinating here is how the American legal and medical system handles a pharmaceutical shortage of this magnitude. When the FDA officially declares a drug to be in shortage, it triggers a specific legal loophole.
The compounding pharmacies.
Exactly. Specialized facilities called compounding pharmacies are legally allowed to step in and manufacture bespoke, generic versions of those patented drugs to meet the public need.
A glaring, dangerous issue with this loophole.
Right. These compounding pharmacies couldn't get their hands on the pure pharmaceutical-grade active ingredients directly from Novo Nordisk or Eli Lilly because those companies guard their supply fiercely. So to meet the crushing consumer demand, many of these compounding facilities started sourcing less regulated alternative chemical versions. Most notably, they started using salt forms of the drug, like semaglutide sodium.
Why is a salt form bad? It's the same base name, right?
It shares the name, but chemically it is not the same molecule.
Yeah.
Semaglutide sodium is intended strictly for laboratory research, not for human injection. When you alter the molecule into a salt, it changes how it dissolves and how the human body absorbs it.
That sounds risky.
It's very risky. It has never been tested in rigorous human clinical trials, meaning the safety profile, the correct dosing, and the potential side effects are a complete unknown. It got so widespread and so dangerous that the FDA had to issue official urgent warnings to the public about the severe risks of taking these compounded, unverified salt versions.
Fortunately, the manufacturing side is finally catching up to the cultural phenomenon. By early 2025, the massive multi-billion-dollar factory build-outs by Novo Nordisk and Eli Lilly finally came online, and the official FDA shortages for both tirzepatide and semaglutide were officially resolved.
Which is a huge relief for patients.
Absolutely. But the science hasn't stopped for a single second. The next generation of these drugs is already in late-stage trials. Eli Lilly is deep into testing a drug called retatrutide. Remember how tirzepatide was a dual agonist? Retatrutide is a triple agonist. It targets GLP-1, GIP, and it adds our old friend glucagon back into the mix. Early data shows it might be even more powerful. And they're also developing orforglipron, which changes the game entirely because it isn't a one-way injection at all. It's a small molecule oral pill that manages to survive the brutal environment of the human digestive tract.
It is genuinely incredible how fast this field is moving now. If we synthesize the core lessons from this entire scientific journey for you, three main takeaways really stand out. First is the sheer power of creative, divergent science.
Looking in the weird places.
Exactly. Solving the obesity puzzle required scientists to look outside the human body entirely to the bizarre biology of anglerfish and the raw venom of Gila monsters, while simultaneously utilizing the absolute cutting edge of molecular engineering, like manipulating blood proteins with fatty acids.
The second massive takeaway is that timing is absolutely everything in medicine. Novo Nordisk succeeded as a business largely because enough time had passed since the fen-phen disaster of the late 90s. The public and the regulators were finally ready to trust a weight loss medication again. And TikTok arrived at the exact perfect cultural moment to market a pharmaceutical breakthrough like a viral consumer phenomenon.
And the third takeaway is the fundamental industry shift we are witnessing.
Yeah.
The staggering, unprecedented financial success of the GLP-1 class has entirely rewritten the pharmaceutical industry's appetite for risk. Almost every major pharma company on Earth is now heavily investing billions in metabolic and obesity treatments, which is something they flat out refused to do just 2 decades ago.
It really is a complete paradigm shift in how we treat the human body. Thank you so much for joining us on this deep dive today. From the DNA of deep-sea fish to the venom of desert lizards, all the way to billion-view hashtags and supply chain drama, we hope this conversation provided plenty of aha moments and gave you a really clear, deep understanding of the fascinating science hiding behind all those daily headlines.
And this raises an important question to leave you with, something to mull over as you go about your day. We discussed how these GLP-1 drugs work by traveling deep into the brainstem and the hypothalamus, signaling fullness and effectively silencing what patients describe as food noise, that constant, overwhelming evolutionary urge to seek out and consume calories.
Yeah.
But if these drugs can so fundamentally rewire the brain's reward centers and craving pathways when it comes to food, what else could they quiet? Could the exact same biological mechanism that stops someone from craving a late-night snack eventually be used to treat other compulsive behaviors, other physical addictions, or destructive cravings? It is certainly something to consider as this extraordinary science continues to evolve.